go over, today we're going to finish chapter 6 and begin chapter 7. All right? The last part of chapter 6 involves enzymes and enzyme regulation. All right? We know enzymes are biological catalysts, and we went over this picture before the exam. So last week, we went over this picture, this diagram about enzyme activity. All right? Talked about the fact that enzymes are dependent upon their shape. All right? The shape of the enzyme just allows for a specific reaction between it and its substrate. All right? And any change in enzyme shape will affect enzyme activity. Yes? Okay. So we went over this picture. We have difference in substrates to products. The enzyme is not broken down. It is actually recycled and combined more another set of substrates. What happens when I change the optimal temperature, or I change the temperature or pH of a solution or whatever, a reaction where an enzyme is present? What happens to an enzyme's activity? Mike? It stops. Does it stop? Does it change? Yes or no? What's that? The proteins do something, yes, not dissociate, but denature. Okay? So when you change, enzymes have evolved over time to have an optimal pH and an optimal temperature. Okay? So an enzyme's activity, excuse me, depending upon which enzyme we're talking about, where it comes from, etc., will depend upon its origin and has an optimal pH and an optimal temperature. All right? On your exam, there was a question about sulfur bacteria that's found in geothermal vents, right? And they're allowed to live there because they have specialized lipids in their plasma membrane. They would also require what else? Specialized enzymes, right? Because those enzymes would have to be able to work at optimal pH and optimal temperature. They would have to work at 80 degrees Celsius. All right? Those enzymes would not work as effectively as if they were at 37 degrees Celsius. And your enzymes that are in your body that work at 37 degrees Celsius don't work the same as at heightened temperatures. Yes, there's an element of denaturation that could happen, a change in the enzyme shape, right? But it just also will affect their activity. All right? So this is a graph showing optimal temperature for a typical human enzyme, which is about 37 degrees Celsius, your body temperature, right? So your enzymes work most efficiently at that temperature. All right? They've been optimized for that temperature and the pH of your body. Optimal temperature for an enzyme for a thermophilic bacteria or a thermophilic archaea is different. It's not to say that I don't have the same or similar enzymes. All right? So you can have a DNA polymerase enzyme. DNA polymerase synthesizes DNA, what replicates DNA. Bacterial enzymes have it. Archaea has it, or has a version of it, as do human cells. Okay? Years and years ago, someone came up with the idea that a thermophilic bacteria that lives at 80 degrees Celsius, you have that enzyme, we can utilize it to replicate DNA. How many of you know what PCR is? 2? 3? You really have to work on your high school biology teachers. Okay? Polymerase chain reaction is the ability to, in vitro, in the lab, replicate and synthesize parts of DNA. And they use that enzyme from those thermophilic bacteria that can withstand that high temperatures to utilize that procedure. All right? We won't go over the procedure. It's a little more than we need to know. But the point is, it's a characteristic of the enzyme itself, the properties of the enzyme that allows it to work that way. pH is also important. Optimal pH for pepsin, which is in the stomach. What's the pH of the stomach? Acidic or basic? Very acidic, okay? So pepsin only works in an acidic environment, right? At 37 degrees, but at a very low pH. Trypsin, which is an intestinal enzyme, right, still in the digestive system, closely related, but as food moves from the stomach to the intestine, there's a change in pH. pH goes from about 1 to about 7.4, right, closer to 8, and then now trypsin can work and break down food, all right? Pepsin doesn't work as well. It gets inactivated when it leaves the stomach into the intestine. And trypsin in the stomach will be denatured and broken down. Questions on optimal pH, optimal temperature. Ways of regulating enzymes even in the human body in different areas. Okay? And each enzyme protein has its optimal, meaning optimal, I mean its most high activity or its greatest activity at that optimal temperature and pH. Other ways that enzymes are regulated. Cofactors. Some enzymes require helpers. All right? They have to have something else that binds to the enzyme that could makes it activity more. All right? Either changes its shape so it can bind to substrate, or increases the affinity, meaning its ability to bind the substrate. It changes the enzyme's activity in such a way. All right? Cofactors may be inorganic or organic. Inorganic cofactors such as iron, you have other vitamins, molybdenum, etc. Right? Inorganic. Or they may be organic. Right? Organic cofactors are called coenzymes. How many of you have heard of coenzyme Q? What it is? You can take that over, they sell it as a dietary supplement. Coenzyme A, right? That's already found in your body, right? So it's an organic cofactor, okay, that will bind an enzyme and give it its more activity. And coenzymes include vitamins A, D, E, K, all the B complex vitamins, etc. Okay? And so vitamins are coenzymes that are organic cofactors. So cofactors enhance the ability of an enzyme or increase the enzyme's activity. On the flip side, you can also inhibit the action of an enzyme. All right? We have two different ways to inhibit the action of an enzyme. How can we inhibit the action of an enzyme? Okay, change its shape. Okay, so we could change its shape. What else can we do? Which will affect what? Well, you change its shape, it's going to affect its active site, and basically it's going to prevent or change the way it binds its substrate. Okay, so two ways to do this is what we call competitive or non-competitive inhibition. All right. Competitive inhibition denotes a competition. Two things are going to compete for one thing. Okay? Two things, meaning substrate or non-substrate or inhibitor, are going to compete to bind the active site. All right? So you have substrate for an enzyme, and then you have its inhibitor. All right? So here's our enzyme. This being the active site. Our substrate's going to fit in here, right? But if we have an inhibitor that binds in this process, binds the active site, that prevents the substrate from binding to the active site, that is a competitive inhibition. There's a direct competition for where the substrate binds, right? A direct competition for the active site. prevents substrate binding. A non-competitive inhibition, okay, means that the inhibitor, does it bind the active site? Yes or no? No, it binds somewhere else. So here we'll do this. Draw back our active site here. Here's our active site. Now, non-competitive inhibition. Going to have your non-competitive inhibitor bind somewhere else. Okay? Non-competitive inhibitor binds the enzyme somewhere else other than the active site, but does affect the enzyme's ability to bind the substrate. Now it doesn't bind as well. All right, so this doesn't bind. The active site is not the site of binding in non-competitive inhibition. All right? So, competitive inhibitors bind to the active site, non-competitive inhibitors bind to another part of the enzyme itself. All right? Making the active site less effective or making the affinity for the substrates less effective. These can be pesticides, antibiotics. Antibiotics. I'm currently on a lot of antibiotics. Okay? Actinomycin D is a type of antibiotic that specifically targets bacterial ribosomes. Okay? Why do we want to target bacterial ribosomes and just not ribosomes in general?
If you attack the bacterial ribosomes, then the ribosomes will be creating more proteins to make more bacteria.
And particular bacterial ribosomes and not your own, right? So we're going to target the bacteria and not your cells because we want your cells to keep living but not the bacteria. All right. Now, ribosomes have enzymatic activity. They build proteins. All right. We target that ribosome. It changes its affinity for producing that building proteins, and then that isn't a non-competitive inhibition. It's an inhibitor. Okay? Here's a diagram showing your competitive inhibition versus non-competitive inhibition. Better diagram than I can draw, but you get the idea. Questions on competitive versus non-competitive inhibition? Now, so we know enzymes are proteins. They're coded by genes. All right? Changes in genes lead to changes in proteins. Changes in proteins lead to changes in structure. All right? Change in the amino acid sequence leads to a change in structure. Because shape and structure is so important in proteins and especially enzymes, change in structure can lead to a change in activity. If that activity is beneficial, awesome. That is passed on to the next generation. If that change in activity is not beneficial or not just bad in general, it doesn't get passed on. Those cells don't live. Give me an example of this type of evolution. Last week, I was in the hospital for my elbow infection. First day I went in, they gave me antibiotics IV antibiotics, Ancef, and then sent me home with an oral medication, penicillin. Okay, I was in here on Wednesday and it was all red still, right? Last week. And on that Wednesday going home, I realized that the infection was still spreading. Why was the infection still spreading?
Because the infection had evolved, so it wasn't, the penicillin wasn't working.
So the penicillin wasn't working. Okay? The way penicillin, that antibiotic, works, it actually punches holes in or prevents the production of cell walls of bacteria. Okay? But penicillin-resistant bacteria have developed enzymes that don't stop or prevent or break down penicillin, or they still work in the presence of penicillin. So, methicillin-resistant Staph aureus, MRSA, is an example of evolution that's occurring in response to mutations. Antibiotic-resistant bacteria. Multiple drug-resistant chemotherapy or cancers that are resistant to chemotherapies is an example of evolution that's occurring on a microcosm scale. All right, real time is happening right there. All right, questions on the evolution of enzymes, and that's related to enzymes, also related to other stuff.
Like the ability to digest lactose, for example, of this. But I know like people in the past like that was not something that would happen before.
Yes and no, because we've always had mammalian development has always required that mothers breastfeed their children, right? So they always have the ability. Children usually have the ability to break down lactose, but now that's been flipped to us consuming cow's milk, and that's a different type of lactose. So yes and no is the answer to that. So we've always had the ability to break down lactose that we've had consuming on our own, but as we've moved away from towards a cow-based diet, that has altered things. So when we talk about regulation of enzyme activity and tie this into what we talk about metabolism, how does regulating an enzyme relate to controlling metabolism? Well, metabolism is a collection of reactions, correct? Yes? We have catabolic and anabolic reactions. And those reactions are catalyzed by what? Jessica? Enzymes. All right? So if a cell needs to produce something more, it can or it needs to control a reaction that's happening, a metabolic reaction that's happening in the cell. Instead of controlling how much of a substrate is there or how much of a product is there, the switch or the place that you really want to control it is at the level of the enzyme activity. So controlling metabolism is directly related to controlling or regulating enzymatic activity. All right? How do we do this? Well, this is a process called allosteric regulation. All right? So allosteric regulation just means a way of regulating, inhibiting, or enhancing an enzyme's activity. Inhibiting an enzyme activity could be competitive or non-competitive inhibition, and enhancing an activity could be the presence or absence of cofactors. Okay? So the presence or absence or cofactors will either increase or decrease the activity of an enzyme, and then whether or not competitive or non-competitive inhibition occurs. So allosteric regulation occurs when a regulatory molecule, whatever it might be, a cofactor or inhibitor, binds to a protein at one site and affects the protein's function at another site. What type of inhibition are we talking about here? What type of inhibition is an allosteric regulation? Non-competitive. All right? A competitive inhibition is not allosteric. All right? So an important part of allosteric regulation is that the binding of that inhibitor or substance is not at the active site, but it does control the action at the active site. So, allosteric activation inhibition, most regulator enzymes in this fashion are made from polypeptide subunits. Each enzyme has active and inactive forms. Okay? So, in the presence of an allosteric activator, the enzyme is bound to the activator and then will perform an action. Okay? And the allosteric inhibitor, it won't perform an action. Okay? So in this case, an example, the binding of an activator stabilizes the active form and allows the enzyme to do its action. Right? Opposite to that, the binding of inhibitor stabilizes the inactive form and prevents the enzyme from doing its action. All right? Allosteric regulation. Here's a diagrammatic example. All right? So here's your allosteric enzyme of 4 different subunits, one of 4. Okay? And there's 4 regulatory sites on there. An activator binds. What occurs? That stabilizes the active form. Now what can happen from here? Here on, what's going to happen at each one of these active sites? The substrate's going to bind. Okay? Is this a completely active form? Yes or no? How many regulatory sites are on that enzyme? 4. Do you think, Beth, optimal activity will occur when only one of those sites is bound? No. Optimal activity will occur when all 4 of those regulatory sites are bound by an activator. Now, all 4 active sites can bind the substrate, and optimal or highest activity will occur. All right. B, cooperativity. It's another type of allosteric regulation. All right. If substrate binds, right, you have an inactive form, binds to one active site, changes this formation of the other active sites, makes it more active. So the binding of one substrate, right, enhances the ability of the enzyme to bind the other substrates. Why is this allosteric regulation? Question mark. If the substrate is binding an active site, why is this considered allosteric regulation? Because we just said allosteric regulation requires binding of a substance that's not the active site. Why is this allosteric regulation?
Still activates the other 3.
It activates the other 3 active sites. Okay? So this is allosteric regulation not for this particular subunit, but for the other 3. Right? That's going to enhance the binding of a substrate to the other 3 sites. Okay? And then finally down here you have the formation of an inhibitor. Right? The inhibitor binds to this. In this case, this enzyme on A right here has both the regulatory site can be bound by an activator or an inhibitor. Okay? That's not the case in all enzymes. Some enzymes are produced in an inactive form, then they bind, they're bound by an activator, and they go to the active form. In other cases, they're produced in an active form, an inhibitor can bind, and now it goes to the inactive form. It's either on or off. In this case, they're showing you that an activator or an inhibitor can bind to the regulatory site and shutting it on or off. Okay? The concept is still the same. Okay? There's a regulatory site away from the active site that is binding a substance or molecule and regulating its activity. Questions on allosteric regulation? This is an easy way for the cell to regulate enzymatic activity. Yes. Right? So if we remove the presence of an inhibitor, okay, from the cell, from the cytoplasm, you remove the inhibitor or inhibit the inhibitor, that allows the enzyme to do its activity, right? If we remove an activator, that will help prevent the enzyme from doing its activity. Okay, so this is a quick way for cells to regulate the activity of an enzyme without having to get rid of the whole enzyme from the cell and then produce more. Okay, that's inefficient. This is a more efficient process. So then we have the cooperativity, right? That can amplify enzyme activity. A little right up. And then finally we have another type of inhibition, feedback inhibition. Okay, this is very important in regulating homeostasis in cells and regulating activity of cells, regulating activity of metabolic reactions. Okay, the idea is go from substrate to product. It's inefficient to produce more product than a cell actually needs. Yes? Because if a cell needs only a little bit, why produce a lot? That's a waste of resources. So in order to prevent this from happening, you have what's called feedback inhibition. The end product of a reaction regulates the enzyme that's catalyzing that reaction and prevents it from continuing to produce the product. Okay? So it's a way of efficiency and making the most of. Here's feedback inhibition. Okay? Here's this. This is using isoleucine. Isoleucine levels in the cell go down. The enzyme that produces isoleucine becomes active. Okay? Threonine, which is another amino acid, binds to the active site. Right? This enzyme we're talking about is threonine deaminase. It's going to take threonine and through a series of steps produce another amino acid, isoleucine. Okay? So you go from threonine binds the active site because low levels of isoleucine are present. Low isoleucine, the active site is not bound by isoleucine of the enzyme. It can bind threonine and then starts this process to produce isoleucine. What is isoleucine in this case? It is also the what type of inhibitor? What type of inhibitor? Non-competitive inhibitor for the threonine deaminase enzyme. So as these levels go up, more of the isoleucine's more of the inhibitor to this enzyme is available, binds the active site or binds the allosteric site and prevents the enzyme from binding more threonine. What's the purpose? It helps regulate the levels of threonine at a certain level and the levels of isoleucine at its optimal level. Right? Feedback inhibition. The result of the process is feeding back on the activity of the enzyme. All right, so right here. Questions on feedback inhibition? This is an important concept. As we move on from here, regulation of enzymes is an important concept to understand. We're going to go through glycolysis and cellular respiration over the next week. All right? These processes, these reactions are regulated by enzymes that are important in how this is regulated. We're going to go through the breakdown of the metabolism of glucose in bacteria, all right, or lactose in bacteria. Understanding allosteric regulation will help you understand that process much easier or much more readily. Right? You'll get that quicker if you understand that. Enzymes are localized in certain structures depending upon where they act. Enzymes involved in cellular respiration and the electron transport system are probably found where? Are they found in the ribosome? All of you should be saying no. Okay. All right. Are proteins involved in enzymes involved in protein synthesis probably found in the nucleus? No. Where they act is where they're found. Okay? So enzymes involved in the electron transport system are found in the mitochondria. All right? So you have specific organelles where they're found, cellular respiration, mitochondria, etc. All right? Some enzymes act as structural components of membranes. You will have enzymes that are studded in the membrane. All right, so integral membrane proteins can have enzymatic function. They are mainly related to cellular signaling. Questions on enzymes, enzymatic regulation. That ends chapter 6. Okay? And then for chapter 7, we're going to talk about how a cell produces energy. And by producing energy, I'm talking about producing what? Specifically ATP. All right? Energy flows through a system. We know that the law of thermodynamics states that first law says what? Energy cannot be created or destroyed. And then the second law of thermodynamics states that what? Energy transfers and there's a loss of heat to the system, increasing the disorder of the universe or entropy. Okay? Yes, we're going to talk about how energy flows from one form to another through a cell. All right? That is in broken down cellular respiration. Okay? So, photosynthesis, where does all energy eventually, what's the source of all energy? From the sun. All right, so sunlight, photosynthetic organisms produce oxygen and also store energy from light in the form of glucose, and then other consumers will utilize that glucose, that cellular stored chemical energy to produce ATP. Okay. That's an energy flow. Light energy comes into the ecosystem. Photosynthesis occurs in chloroplasts within bacteria and other photosynthetic organisms, produces organic molecules, whatever it might be. We're going to be concerned with glucose because that's the one we're really concerned with, and produces oxygen. Other consumers utilize the oxygen and glucose in a reaction to transform the energy stored in those molecules, in the organic molecules, into ATP, which then the cell does its work, producing CO2 and water, okay, which then is recycled back into organic molecules. All right? So, we have catabolic pathways. What does catabolic mean? Break things down. Glycolysis, the Krebs cycle, and the electron transport system, the three parts of cellular respiration, are catabolic reactions, is a catabolic process altogether. We're going to break down organic molecules, glucose, in the presence of oxygen, and harvest that chemical energy and store it in the form of ATP. Okay? What are these pathways? Okay? There's two separate pathways where this can occur. We occur in fermentation. I'm going to talk about that. And aerobic respiration. Okay? Fermentation is an anaerobic process. What does anaerobic mean, Amy Jo? No oxygen. All right? So oxygen is not utilized, but what's still occurring, partial sugars, organic molecules are broken down, and usually CO2 is produced. Okay? Aerobic respiration, much more efficient. Glucose molecules are broken down in the presence of oxygen, yields ATP. Both fermentation and aerobic respiration produce ATP. Okay? There's a type of anaerobic respiration which is similar to aerobic respiration, but does not depend upon oxygen. Instead, there is another electron acceptor at the end. All right, some bacteria do anaerobic respiration, break down glucose, not in the presence of oxygen, but maybe in the presence of sulfur, and dump the electron on sulfur at the end. Okay? That is a different process than fermentation. Okay? So really, three different processes, ATP. We are concerned with cellular respiration first because it is the one we use. It includes both aerobic and anaerobic pathways, right, or parts of the whole system. Some are aerobic, some are anaerobic. We'll specifically talk about glucose, but you should understand that fats, proteins, nucleic acids, can be consumed as fuel. They can and will be broken down and feed into the system that we're going to talk about at different spots. All right, and they can be and will be used as fuel. But we're going to start with glucose. So here's the reaction, overall reaction. C6H12O6. I won't do it, right? In the presence of oxygen, plus 6 oxygen, right, yields 6 CO2, 6 waters, and energy. Okay? And that can be anywhere between 30 to 34 ATP. Okay? Energy in the form of ATP, and some will be released in heat. That is your overall reaction for the process of cellular respiration. Now, is that reaction oversimplified? Yes, very much so. There are several steps that occur within that reaction. We will go over all of them when we go through. All right? It is important to understand what these reactions are. They are typically a transfer of electrons. All right? These reactions are going to move an electron from glucose, eventually from glucose, all the way over to oxygen, which is where you're going to end up with water. All right? So these are what we call oxidation reduction reactions. Well, all that is movement of electrons. And you remember, I'm sure you've seen this, your oil rig. Okay? Oxidation is losing electrons. That is what it means. So if something's being oxidized, it is losing an electron. That electron has to go somewhere, right? Yes? It doesn't just fly off into space. Matter can neither be created nor destroyed. So it just doesn't go anywhere, doesn't get expelled. Okay? There has to be something that's going to take up that electron. So reduction is gaining. That electron is going to go somewhere. Wherever that electron goes, that substance is being reduced. Okay? These reactions are coupled. Something cannot be oxidized without something else being reduced. Okay? So, oxidation is. So the principle, oxidation, the substance loses electrons or is oxidized. In reduction, it's gaining electrons or is reduced. So, this is showing the couple. Okay? Here's a hint. This is an inorganic reaction that you're looking at. Okay? If we look at an organic reaction such as C6H12O6 plus oxygen yields CO2 and H2O, if you follow the hydrogens, you follow where the electrons are going. Okay? So, C6H12O6 plus oxygen yields CO2 plus H2O. What substance, where is the oxidation reaction? Oxidation is losing. Follow the hydrogens. What substance is being oxidized? Glucose is being oxidized to what? So follow the hydrogens. This is the hydrogens. Oxidation is losing. What on the other side of the yield sign of the arrow is losing hydrogens? Carbon dioxide. Okay? So glucose is going to be oxidized to carbon dioxide. Then on the flip side of that, what is being reduced? Where are the electrons going? Where are those hydrogens going? They're going to water. So oxygen is going to be reduced to water. Okay? This is a reduction. This is an oxidation. Coupled reaction. Okay? So in organic reactions like this, especially in the overall reaction for cellular respiration, follow the hydrogens. Where the hydrogens go, electrons, that's really where your electrons are going. Okay? It's a basic rule of thumb. It doesn't apply, it's harder to apply it to, obviously you can't apply it to inorganic reactions. Basic rule of thumb, if you follow the hydrogens, that's where things are going.
So wherever the hydrogen is going, usually the one that's reduced?
Yes.
Okay, thank you.
All right. Other terminology, your electron donor is called the reducing agent. The electron acceptor is your oxidizing reagent. So what's the electron acceptor in the C6H12O plus O6 plus O2 yields CO2 plus H2O? What's your electron acceptor? What is being reduced? Well, you're being reduced to water, but what is the starting material that is being reduced? Oxygen. So your oxygen is your oxidizing agent, and your reducing agent is the electron donor. Where are those electrons coming from? Glucose. So this tends to confuse a lot of students, this terminology. My point, if you follow where the electrons are going, what is being reduced, what is being oxidized, where are the electrons coming from, where are they going? That's more important than knowing this exact terminology. Because the reducing agent, okay, which is glucose, is going to be your electron donor. But what glucose is being what? Oxidized, right? It's going to lose electrons. So this is the terminology is flipped. Okay? Do not get confused by that. All right? Do not let this terminology confuse you. The important is to understand where the electrons are coming from and where they are going, and that will help keep things straight. Okay? Questions on that? Some redox reactions do not transfer electrons, but change the electron sharing and covalent bonds, sharing where the electrons are. All right, and that's transfer. All right, so here's your becoming oxidized reaction, right? And you see the methane and oxygen, they're not changing necessarily where, they're not transferring electrons, but it's transferring where they are. Okay? What's happened is you've actually flipped where the sharing of electrons are. Okay? Questions on oxidation? All right, so redox reactions tend to move electrons closer to electronegative atoms. What does electronegative mean? They are found in polar molecules. They have a high affinity for electrons. Right? They like them. So when electrons are being moved, they tend to attract them. So it's only intuitive that when a redox reaction occurs, it moves it towards electronegative atoms like oxygen. And then when that occurs, energy. All right, we'll stop there.